Go and thank you all for participating. I'm not sure if any of you have participated before in one of these, so I'll go through like a quick kind of orientation, but we want to spend most of the time talking to Hakeem about his Ames page and his plans. The idea here is to get some friendly advice for Hakeem. Looks like he's frozen. Hopefully he's still there. It's for him going through his grant. So we tried these a bunch of different ways. We did mock study sections, which were brutal, and this is meant to be a little bit more interactive and helpful to Hakeem. Hakeem, are you there?
Yeah, can you hear me?
I can hear you.
Yeah.
Your video's frozen, but I just want to make sure that
Okay.
you're on.
Okay.
So we'll go around, and we've all done reviews before to summarize sort of what you think and some suggestions for Hakeem. We do have 4 reviewers, and I want to make sure there's time for back and forth and discussion. Julianne, is it recording? Yeah, so Hakeem, you can be in the moment and interacting with the other faculty on the call, and then you can review.
Okay.
If you feel like you need to take notes contemporaneously, that's great. Also, so we're gonna go around. I'm just gonna go by the Hollywood Squares, so I can keep track. So Amanda, I'll ask you to go first, then Matt, then Jessica, then Diane. We'll go in that order. But before any of that, what I always have people do is Hakeem in 3 to 5 minutes at the most, so
Right.
quickly, just summarize what your intent is with this. And that's a good way, especially if there's somewhat of a disconnect between what you're trying to communicate and what's written. It's a good way to get the reviewers kind of oriented on what you're trying to do. And if you could lead off by what's the grant mechanism, and if you have a particular study section that you're going to, any specific information about it. So you, sir, are up, Dr. Lawal.
All right. Thank you. So can everybody see me yet?
Your video's frozen for me.
Oh,
Yeah, I think
no.
everyone's. Yeah. It's okay. You're
Okay.
smiling at least.
Good, good. That's what
It
I
could
want to
be worse.
remember.
It could be
The
worse.
smiling guy. Yeah. So I actually have a slide to share.
Also, I don't organize them through June.
That was kind of would work. So whatever works for the panel is fine with me.
You can share it. Go ahead and share it while you're talking.
Okay. Okay, great. So let me start with the mechanism itself. So let's see how this works. Sorry guys, screen share, panel window. Meeting. Trying to pull up that. Sorry, let's just have a. I have a actually. You know what? I'll just drop this link in the chat. I wanted to share with you the mechanism itself. So I always joke that this mechanism has my name written on it because it's the R1 reward R1. So it's the NIH NIH loves acronyms and snazzy stuff. So it's called reward REWARD, and so the idea is to essentially support PIs like myself that are at institutions that are either not primarily research focused or otherwise are doing a lot of EDI work, a lot of non-research work when they would rather be doing research. I was like, whoa, this is me, So the NIH put out this called the first study section. The first submission was last October, or either September or October. So this is going to be the second one was in January, and then the third one is the one I want to go for. So I want to just give the committee just an idea of that. So it's a very new mechanism of the R1. It's designed for investigators who want to do high octane research. They have programs that can support it, but for some reason, through their work with service and EDI and student mentoring training, they just haven't had that bandwidth or the time or whatever, what have you. So and then another quick information for the panel. I have submitted R1s before. I actually gave up about 3 years ago. And so Rob might remember some of my questions from the gene meetings. It was
Mhm.
kind of like tinged with frustration. That's why I was like, I'm done with this thing. So one of my colleagues at Delft actually advised me, why don't you go to NSF? And so I did have success at NSF. But when I saw this new mechanism at the NIH, I decided to go for it. Okay, so that's kind of by way of the mechanism itself. And then let me try to share my screen just really quickly. I'll take no more than 2 minutes or so to show you kind of like, let's see. And I'm really sorry, I'm not so familiar with, I'm not so familiar with upload from my computer.
Teams. So there should be a
Yeah.
share button up and to the right next to the leave button.
Okay.
Yeah.
Okay. Oh, yeah. So I see show participants, more actions. Oh, I see this. Close share tray. Sorry, guys. The time is really precious. Screen share. Okay, I'll do this one. Screen share. Oh, desktop. I found it. And then, oh yeah, here we go. Can everybody see my screen?
Yes. So can you go
Okay,
briskly?
perfect. So
you go briskly?
Yeah,
through
no,
these?
I'm just gonna show you. Yeah, just a few things, just 2 slides on this screen is what I want to show you. So did you see Parkinson's disease, many facets?
Hakeem, yeah. I'm not sure we have the bandwidth with your connection. Are you still there?
Okay, okay.
So you might not be able to share and
Here.
speak
Let
and
me.
use your video at the same time.
Okay.
So just verbally,
I will.
I mean, the group is pretty established in the area. So just give a high level verbal sort of accounting of what the grant's for. Okay.
No. So the grant is to
Hey Hakeem, sorry, you'll have to stop
we
sharing. I don't think we have the bandwidth for you to share. And
okay, sorry about that. Let me stop sharing. Have I stopped? Am I still sharing?
I still see it. Yeah.
Okay,
Here.
let me write it. Oh, yeah, you're right. So yeah,
Let me kick you out.
Stop. Okay. Yeah. Let me log back out because it's not letting me stop. Let me log in and log out. Okay, I'll be right back. In 1 quick second.
So once Hakeem gets back, actually Jessica's back, but and everyone's here. Okay, so once Hakeem gets back, we'll do this sort of like a regular study section, only in the sense that if you have something similar to say someone who went before you, and again, I'm just going to go around the clock, just underscore it. You don't have to spend a whole lot of time. So try to get him as many different pieces of information as possible. But part of it's interactive, so don't feel like you have to do your report, right, your presentation or your critique, and then he can talk. Because if you have questions, you're trying to clarify something or something wasn't clear, it's very helpful to the person submitting the grant to just deal with it right then while you're talking about it. And he can certainly go back and look. And if you have written comments, it's not 100% necessary, but if you do, that would be great also. But we kind of need Hakeem. Hey, Diane, do you have a question?
Yeah, so if I made comments and I marked up the PDF, can I just email it to him? I didn't know if this was, okay, I'll do that.
Yeah, absolutely. This is just we're trying to facilitate him being able to pick your brain. I know you probably see him. Well, I don't know actually how the neuroscience Cobri folks are still, if you guys still have contact with each other. But this is, whatever you have, send it to him so he can try to digest it as he's pulling the R21 together. I'm sorry, the R01 together. All right, Hakeem.
Yes, can everybody hear me now?
Much better.
Okay, all right, good. Okay, so yeah, so the brief gist of the grant is that I have these we have this compound in the lab, which actually is the basis of my just concluded INBRE pilot. And so we're testing a neuroprotective mechanism for Parkinson's disease. That's the long story short. And we want to do two ways with the R1. The first one is just purely mechanistic. So the question is, can we use dopamine release as a way to treat Parkinson's? Can we manipulate dopamine release as a way to treat Parkinson's? That's the kind of like very nerdy way to look at it. But the more like translational way to think about it is, can we have a new anti-Parkinson's drug? Okay. And both questions lead to the same answer, which is, if we can target the release of dopamine, at least selectively, as a way to give patients, so in a way that L-DOPA would not be able to do, for example, then that would be a great thing. Mechanistically, if we can actually use ways to, effect a better, what we like to call physiological release of dopamine in striatum, we think that that would actually overcome some of the side effects that Sinemet is known to have, right? So that's kind of the spirit of the grant, if you will. The nuts and bolts of it is taking a bunch of different models that we have in the lab. So during my postdoc days, I did a lot of work with rotenone. This is a compound that sadly is still being used in some spaces as a pesticide, but it's known to cause at least to be let me choose my words carefully, the risk factor for sporadic Parkinson's disease. But it's very useful for us in the lab because depending on your hands, it can be quite reproducible to kill dopaminergic neurons, and then subsequently to look at non-model or model symptoms of the disease. So some of the aims are geared squarely toward this environmental model. And then we choose another one where we focus solely on the familial or the genetic Parkinson. So we have alpha-synuclein model in the lab that we're very kind of happy that it works very nicely. I'm happy to talk about that in Q&A. And then the third part is to combine both together, right? In each of these systems, we're going to be testing these compound that we have called dacarbazine. And then the other kind of not so neat story, but we kind of managed to find a way around it, is that dacarbazine is actually an anti-cancer drug. It's a cancer, it's a chemotherapeutic. And so what we've done which again, for the uninitiated, it means it's toxic, basically, because that's part of the deal, right? You want to kill cancer cells by killing them. So what we've done is that we found a non-toxic analog of that compound. So we think that there is an active moiety that can actually promote dopamine release. So we've kind of taken that, and we're using that a lot in the aims. But we've actually taken it a step further, which is aim 3, and I'd actually appreciate feedback on that. That we were working with synthetic chemists. So this new guy at Dell State, he does like AI stuff, which is way over my head, but it tells me that he's able to use these large language models to predict new ways to rotate the compound that would make it a better agonist for the target of dopamine release that we're looking at. So in a nutshell, aim 1 is really just about the model itself in Drosophila. We want to drill down on what we know already, which is that this compound is able to ameliorate models of Parkinson's in our hands. Aim 2 is really focused on non-model symptoms of Parkinson's. So we have, and it will be, you put yourself in trouble these days if you don't talk about non-model symptoms of Parkinson's, because it's a very big part of it. And for us, it's very fortuitous because we've been moving in that direction anyway. So aim 2 really kind of just drills in on that. So we're using sleep in our own case. And then aim 3 is where things get very interesting. So we are really looking to move beyond these 2 compounds to develop new ones and then go back and test them for whether we can have the same effects that we've had with the compounds. So I'll stop there, and I think hopefully that's giving some gist.
Yeah,
and I'm
great.
still, yeah, and my camera is still not up, right?
It's okay though,
Oh
Hakeem.
man,
It's fine.
yeah, I worked so hard to make my hair look very nice, you know, for the group.
That's awesome. So we have about 45 minutes and there's 4 presenters. I can send comments. I don't have to present right now unless something comes up, there's a question. So shoot for about 10. So highlight the major questions you have or sort of suggestions you might have for Hakeem, but try to shoot for about 10 minutes each, and that'll give us a little tiny bit of wiggle room for discussion. So Amanda, you're still in my top left corner, so you're up.
Okay. Yeah, so I've actually never done a study section before, so I'm not sure what exactly the format is, but I'm just gonna throw.
You want me? We can let we suggest your own study section. Matt's done reviews. You wanna defer? No? Go for it.
I mean,
say you just
yeah, sure.
do
Maybe
it.
I'll defer just to see what the format is. Like I've got a bunch of feedback, but I'm not sure, if there's a better format to
Okay,
give that
who wants
feedback.
who would like to volunteer for going first? Donald Trump, Jessica, or
I could go first. I could go first.
Diane? Okay, excellent.
Yeah,
Perfect.
I forgot I took my camera off just in case there was a bandwidth issue.
Okay.
Okay, so Hakeem, first off, great that you decided to apply, and this is part of my 10 minutes, so don't worry, Rob. I completely feel the frustration with the whole process and the negativity, and is what it is. So I'm glad you're back in the bandwagon. When we did this for Cobra, one of the things I did not like about the Cobra feedback, it was way too negative, way too, so I'm going to try to make mine not like that. And then the last thing I would say is you have to take what I say with a grain of salt, because clearly Parkinson's is not my thing, right? So I would be the reviewer who the SRO or PO assigns to the grant, who's supposed to be like a lay scientist, who just kind of is a scientist, but isn't in your field. And I guess the tough part about the grant is how do you write it so an expert who is in your field could say it's great, and somebody who's kind of ignorant like me, not really in your field, could also think it's great. It's a tough balance in that. All right, so let me start off with if it's okay, I'd like to start off with your personal statement. So I'll go with the good stuff and then things that I think you should think about. So the good stuff is your funding stuff is good, and you've really highlighted that. You have a nice funding period across many years, and it's hard for a reviewer to say that your research is insignificant, right? So that's great. Your publications also is across many years, and I think I saw some Alzheimer's there, related research with your cholinergic work. So that's great as well. Now, I didn't think I saw any Parkinson's work there, so that would raise a red flag to a reviewer, like, well, why are you doing Parkinson's? You're gonna get this, so there's a negativity, we've hung out, so I hope you can take it. This is just what I think a reviewer would come with. With the 18 publications, a reviewer is just gonna say, you're not that productive. I think I was doing like 3 a year for 5 years, and a reviewer still said I was not that productive. 1 year I had 6 in 1 year. This is the first time I came to UD, and the reviewer said I'm not that productive. So I guess it's something that you could say, but that's something. Now your funding offsets that, but that's something that might come up. I guess my main thing is your personal statement. You have some good stuff in there, but when I read a personal statement, I'm looking for the person to convince me in writing that they're the best person for this grant. And when I see it, I see you have a lot of good things. You want to train underrepresented groups. That's great. You're showing how you have some of that fly skill work, how you've been productive as a grad student, as a postdoc, as a PI. But the way it's arranged, it's not really selling yourself very well. So I would stop mentioning too much your mentors, because at this point, nobody cares about them. And this is a mistake I make a lot, which is you don't give yourself enough credit. you have to tell people, I did all this work, and there were, in my molecular psychiatry publication, there were 15 authors, but I was the first one because I was leading the project. You have to really, every sentence you write, is this selling, am I putting my best foot forward? Is this convincing a reader that I am the best person to lead this project? So in your regular life, you probably shouldn't be like this. But when it comes to the grant, it's me, right? That's it, just me. You're picking this because I am the best person to do this. So that wasn't coming through. I think you need to make that a little more forcefully. And one way to do it is get rid of all of your, just briefly mention, I worked under this, I worked under my postdoc, but everything else, this is how I contributed to this lovely product. All right, so the specific aims, these, again, I'll start off with the good, and then here you really want to take what I say with a grain of salt, because I'm a layperson. Having new compounds to treat Parkinson's is significant. I think you're going to have a tough time finding somebody who says that's not significant. So everybody's going to be on board. This is something worth pursuing. When I read it, I thought you designed a compound, and you were now, tested it out in fly larvae, and now you want to take that further, and that's all good. If that isn't the case, then you want to make it clear you didn't design this compound. This is just a compound that's been used for cancer that you're going to try to use for Parkinson's, so that makes sense. But in my mind, there are two things that really aren't coming through. Why is the fly model the best model to use for the question that you have? So if you think there's something that the fly model can do that mouse models can't, then I think you really have to say this. I'm using the fly model because it can answer questions A, B, and C. I've used the fly model throughout my career, so I'm really the best person to lead this. Now you're looking at alpha-synuclein knock-in, right? So that familial Parkinson's, and you're looking at sleep symptoms and motor symptoms. So you said it, that Parkinson's these days is a big thing about it is non-motor symptoms.
Non-motor symptoms, yeah.
You see, but I didn't know that, right? So when I read it, I'm like, why is this guy looking at sleep stuff? Parkinson's is all motor symptoms, right? I feel, and then you always get negative as a reviewer, right? I feel like you're doing it because you can't look at motor symptoms when that's not the case. You're doing it because it probably does make sense. But that has to be really clear. My second thing is, and then after this, you can ask me questions because I would be done. L-DOPA, which you compare a lot, is bumping up dopamine levels, right? And you have this drug that's, I believe, agonist for VMAT. And so that's also going to bump up dopamine levels. But they're working in the same way, right? L-DOPA is a biochemical way to bump up dopamine, and VMAT, let's say this new compound, is a pharmacological way to bump up dopamine. But I don't see it as different mechanisms. I see it as different ways of doing the exact same thing. You're bumping up dopamine levels. Now, there's a problem with doing that because these cells degenerate anyways, and so L-DOPA becomes ineffective after some time. So when I read it, I felt like you were kind of approaching the bumping up dopamine from a different angle, but it's the same thing. You're increasing dopamine levels. You're not really protecting dopaminergic neurons. And so the big question I had is, well, what's the big advance? This chemical just seems like another way to bump up dopamine. Why not just use L-DOPA? Everyone's been using it forever, so you know all the side effects. So that has to be clear. Like, what's new? Why is this better than L-DOPA? Why should you pursue it? There's some other things, but it's in some PDF documents I could send you. But those were the two main things that stood out to me with your specific aims. I don't know why you're doing it in a fly. You haven't really made a case. And even if everything works out, you have a chemical that does the exact same thing that L-DOPA does. So what's there?
Thank you. That's great. Thank you.
I hope it wasn't too negative.
No. This is amazing. I wish all the reviewers are like you.
Do you have any questions, Hakeem, right away, or you will move on to the next person?
No, I'm taking all the notes. I'll ask Dan questions. Yeah, this is all good stuff. And
Okay.
yeah.
All right. So for me to keep track, I'm still going to go around the racetrack.
I can, yeah.
Yeah. Okay. You good?
Yeah.
Amanda?
Yeah, so I agree with everything that Diane said. I think from the perspective of your personal statement in your biosketch, what's really important for, especially for like these, you know, kind of different mechanisms, is to really be, I think, just very direct in how your application addresses that specific mechanism. So for the reward mechanism, say like, this is what I've been doing, this is how, you know, it's in there, but I think it's okay to be really upfront about it. So I think that was sort of the only comment I had different from Diane's comments on your personal statement and your biosketch. For me, when you were talking about the project, I totally understood. I got it. I thought it was really interesting. None of that background comes through in this Ames page. So I think you have to start it the way you started it verbally. You have to say like, you know, everything, like we're interested in increasing dopamine, and we have this compound that does that, and we've got this preliminary data that's really interesting, and we want to try to understand the mechanism, and we want to try to, you know, what you said about this drug is toxic, but this analog is probably not toxic, and so we want to see if they're working in the same way and if they can both be kind of protective. And I don't have a problem with the fly model per se. I think like it's okay to work in flies, and I think there are lots and lots of reasons why people work in flies, including it's really high throughput, and you can do a lot of these screens kind of quickly. So from my perspective, I'm not sure that, you know, you need to spend a ton of time doing that. I do think that you can take some of the detail out of the Ames and put it into the background, because I really didn't get, like, the background starts off kind of like, this is Parkinson's disease, and then so I also am not a PD researcher, but, you know, then you start talking about this increases dopamine uptake, and I'm like, oh, okay, so what? Like, what does that mean for Parkinson's disease, and how is that clinically relevant, and why? Just a sort of like, you also don't, you know, I think there's a lot of value in saying these are the environmental, you know, there's an environmental exposure issue that causes Parkinson's, and then there's also genetic issues, that we want to look at both of those things individually and together to see, but that didn't necessarily come across in this intro part either. And then I like when Ames have like hypotheses, and only I think your first Aim has like an explicit hypothesis statement, whereas like Ames 2 and 3. I feel like Aim 3B, and I can be told that I'm wrong, I feel like Aim 3B is a lot. Like, I feel that this whole pretty big already, and you could almost leave Aim 3B as a future direction.
Wow.
Yeah, so I think really kind of outlining what the limitations of L-DOPA are, why these drugs address those limitations, and then how that's relevant for Parkinson's. It needs to go kind of a little bit more clearly up front. But I really do think the project's interesting, and I liked how you described it verbally a lot, and I think that there's some good stuff in terms of like the Aims. I think it just needs a little bit stronger upfront background.
Yeah, thank you so much, Amanda. Yeah, I really want to, I thank you so much for talking about Aim 3B, and it's something that hopefully we can have time to come back to. Thank you so much.
I think one of the things, like, just as a suggestion on how to conceptualize some of the comments, it's tough if you haven't been on panels before or done these kinds of reviews before. You just picture, this is what I do, you picture someone who, they're trying to help you, they're a little bit tired, they're kind of, you just have to walk them through the thing so that they know exactly what you're talking about, and they can connect the dots pretty straightforward. It's help them help you is sort of a way to think about it. It's not that they're trying to give you a hard time by giving you critical reviews. A lot of times it's because they didn't get it. And that goes back to, well, how do you present it? So I always picture someone who's knowledgeable, not necessarily in the field directly, but kind of sitting there late at night. Maybe one of their kids is crying and they're just trying to get through the thing, and just make it easy for them to help you. So I think that's a good way to conceptualize comments. Do you have any questions for Amanda?
No, I just want to thank all of you. I wanted to save this to the end, but after Amanda, I just have to say a big thank you, because this is what I've missed for so many years. Honestly,
Yeah,
if
that's right.
if I'd gotten this for with my first R1 submission, I think after my third submission, I would have gotten the grant.
Yeah, right.
Because this is, so thank you. All I appreciate it. Thank you.
Yeah. I think I have found these to be remarkably helpful to people. Myself too, I do these also. So Dr. Butchback, you are up.
Dun, dun, dun. Hold on. I'm setting a timer because we all know that I go on and on and on and on. So, it would help if I would set my timer correctly.
Why isn't your timer set already? This is part of you taking time.
Forget the timer. I messed it up. Anyhow, as most of you know, I'm more of, I mean, I do work on motor system disorders, but I tend to be more lower than upper. So, but this actually was a little nostalgic, because this is actually my first foray into research was looking at the Parkinson's model, actually with rotenone, of all things. Back when I was at Ohio State with working in Don Higgins's lab, who was worked with Tim Greenemeyer ages ago. Yeah, so it's like, it's weird, like we're all how we all somehow intersect. But I mean, overall, I think it's a really good plan overall. And I'll go through. There are a couple of things a couple of specific nitpicky things I'm going to go through. But before that, I want to talk about your biosketch and your personal statement, which actually I really like. I like how you talk about both your scientific goals and your research and your research accomplishments, and also talking about your role as a mentor, especially for underrepresented minority students. I think that's very important, especially at Del State. I know that's a big mission there. The only thing I would suggest is maybe switch the paragraphs. So have your mentoring part be a separate paragraph, not just after the intro. So just take like the first couple sentences in your first paragraph, add that to your research interest paragraph, and then have a separate paragraph talking about your mentoring goals, especially with respect to training the current next generation of underrepresented minority scientists. And I think you need to hype it up a bit more, because I think it's so important. And I also think it's one thing that sets you apart from other researchers. So other than that, I think everything is fine. Everything else in there is fine. But then, so that gets me to the specific aims. And again, overall, I think it's a good plan. It's a very good plan, actually. I do have a couple of specific questions, and one is actually, one of the models you're using is rotenone toxicity. So you may want to talk a little bit in your specific aims about why rotenone is really considered a pharmacologic model for sporadic PD, especially because most of us know rotenone as a complex 1 inhibitor, and many the few of us who've actually done research on the neurotoxicity of rotenone know what it really, that there is a PD-like symptomology. So I think that needs to come through, especially if your study section is going to be broader, not just a PD section. And then you also mentioned that your other model is alpha-synuclein knock-in. Is it wild type alpha-synuclein, or is it a mutated, or is it a PD associated mutant?
So we're using just the wild type.
Synuclein.
Right. So
okay, I would make that very clear in there. That's kind of important, and maybe if you can fit a sentence in there about why ectopic overexpression of alpha-synuclein is believed to cause a PD-like symptom. The other, so I think that's again, it's just minor, relatively minor nitpicky stuff. But and then and one other thing you're talking about how you're measured AICA increasing dopamine uptake in 293 T cells. Is there a particular reason why you use 293T as opposed to, say, a dopaminergic cell on like the N27s?
Yeah, no, that's a very good question. So the answer is no, there's no specific reason. It was actually very fortuitous. I had tried for years to get this assays to work, never worked. And then I was at a conference in Switzerland. I met a guy from Gary Miller who also did some of those environmental Parkinson's. I met a postdoc from his lab there, and I was sharing my frustration. This is not working. And she was like, oh, we have this HEC293 cells for dopaminergic assays. It works great. We love it. I was like, no way. And she's like, yes, way. Like, okay. Yeah, that was it. So it's not.
It's weird because, I think, and again, this is more of a, thinking, why is he using a kidney cell line to look at a dopaminergic neuron? So what you may want to have, if you can, and when you actually write the full proposal and you have some data, you may actually want to have, if you can, show some data that these actually do have, the 293Ts have dopaminergic properties.
They do. Yeah. Okay. Thank you.
Just to really, because it convince a curmudgeon like me that that's a valid model, or propose using N27s as an alternate, which are rat dopaminergic neurons, and I believe they are commercially available. I don't know where though. Other than that, I think, that was really one of my main things. When you talk about the sleep fragmentation patterns, especially in AIM-2, you may want to add a statement in there about that it really describe the sleep abnormalities that PD patients tend to have, and just so we can make that connection of why are you looking at why. In this case, FLY is actually is a really unique, good model for this, from what I've read in your previous work and what I saw at the Delaware Neuroscience Symposium.
Yeah.
So I think just kind of just little pieces to kind of bring, those of us who are not necessarily immersed in this field, but kind of know bits and pieces, just to bring everyone up to the same level. I do agree with Amanda that maybe AIM-3B should be more of a future direction as opposed to part of it, but if that's just my opinion on that. Do you mention anything about? And I know the FLY is a great high throughput model and has many advantages over mammalian models, but you may also want to mention something in your future directions about maybe down the road looking at this in more of a mouse or rat model. Once you have hits from your analog screen or AICA. Testing it in a mammalian system, just to because ultimately that's what you're going to need to do that.
That is correct.
Especially if you want to advance any of these compounds that you're going to be coming up with into preclinical and ultimately clinical trials, you're going to have mammalian data. So I think just at least if you're not going to if you don't want to, and I'm not saying you have to put it in here, but just have like a statement, like again, in future directions, this is what we wanted, this is what we'll do, these are our next steps, and we'll try to find experts who worked in rodent models of PD that can help with experimental design and all those other nitpicky things that that we mouse people tend to have. But all in all, it's a really good idea, and I think it's a good. The reward mechanism this is a really good proposal for this mechanism. So I'll leave it at that, and I think I actually was under 10 minutes for once in my life.
You did amazing. That was awesome.
Yeah.
Perfect. So, Jessica, how are you? I haven't seen you in a while.
Yeah, I'm doing well. So
excellent.
Okay. So take this all with a grain of salt, being that I am the other model organism researcher here, And I was, as you know, recently burnt on an R01 review. Okay, so I may be a little more harsh because of that. I would also say that these, and we talked about this, the study section that you get into with this type of proposal will make or break you. Okay, there are many study sections that this would not be discussed in. So I do know, do you know Tim Mosca at Thomas Jefferson?
Yeah, I know Tim Mosca.
Okay, he would be a good person to talk to because we were talking about study sections and just see which study sections he has had grants successfully funded in. So I think that is really, really key. Productivity, right, is field dependent. I think your productivity is, you know, is fine, provided that you're in the right study section. With the bio sketch, I agree with what everyone has said so far, but I also feel like it would be really great at the end to have a statement, a couple sentences about why your environment is going to enable your success, and you know, why is your environment just so much better and will allow you to succeed. Okay, so I think that one thing, and someone else mentioned this, is that 2 of your aims did not seem hypothesis driven, or maybe they were hypothesis driven, but you didn't explicitly, you know, hit the reviewer over the head with that type of statement. I kind of feel like you need aim 3B, otherwise I don't think it's ambitious enough. However, at the same time, so I'm going to argue both sides here. So at the same time, I feel like aim 3B is a fishing expedition and it's a really high risk. Right. And so they're going to come at you from that, because I've proposed screens before. And at least in my last grant, even though I proposed a screen, they still wanted hypotheses about the screen. So even though it was a screen. I think your preliminary data as far as the transport assay is going to be absolutely key there to show feasibility. I felt like in aim 1, I'm not sure if you could get more into like mechanism at all, but that didn't really come across to me. It more seemed like we have this drug and we're going to test it in these assays, and I just felt like in some way it was kind of lacking in depth there. I know you do some work, and this may be opening a total can of worms or flies, but I felt like your lab has looked at sex differences, and you may not want to get into that here at all, but I was just wondering, were you planning on looking at sex differences or only looking at one sex? Because I know that you've seen differences with different sexes. Yeah. You might have written that, but if so, I missed it. I also, when you talked about rotenone, it completely made sense, but when I read the aims, I think it needs to be addressed more broadly. You know, you said it killed specific neurons, and that's why it was a model, and I didn't get that at all from the aims. And then this may be just me and my personal preferences, but I do like when I'm writing my aims, I like there to be a little more space, because having like small kids or grad students busting into the office, it's often hard to get through one entire page without an interruption. And I feel like if there's a space, you can at least be like, okay, I got through, you know, up to this space before I got this, you know, interruption. And I think just making it as easy as possible for the you know, the reviewers to digest. It's amazing, and again, and that and selling the model system, because you'd really hope that you get a Drosophila reviewer, but I have to say, my last R01, which was not funded, and my last R03 application, which was funded, none of the reviewers were worm reviewers, which seems nuts to me. But you really gotta sell your system, and they're not going to be as familiar with all of the different techniques and the genetic mutants you use. Maybe you'll get one, but probably not more than one. So I would keep that in mind.
I think. No, I was gonna. Do you have more?
I think that's it.
Yeah. And I think for this particular mechanism, have you talked to the program officer like multiple times already? Because this is kind of a different mechanism because of the DEIA component of it. So comments like Jess said about the study section, I bet you go to a special emphasis panel, and then the odds of getting a dyed-in-the-wool Drosophila researcher who's also familiar with Parkinson's becomes small, right? They're going to be more generalist, which means you have to write for a general audience. It gets really difficult because a lot of the specific details, like what Matt was bringing up, you still have to say them because they'll look it up or they'll find it, but you have to engage them like where they are. And it's really tricky with a special emphasis panel because you can't see who's on it. You don't know what their background is. But back to my question, have you talked to the PO yet? Because there's some specifics in the FOA or the NOFO that are a little bit different.
Yeah, no, that's a good question. So, so the answer is yes and no. I mean, the yes part is that they actually visited us at Dell State, a group of them, to talk about this, this mechanism. And then I've also talked to my, at NINDS, I've talked to a program officer that I've worked with in the past, and she recommended that I do this, right? But I've not had additional conversations with them about exactly what you've said, and like what Matt was talking about, and Jessica just did too, like how does all that factor into what I'll be writing? Mm-hmm. If there's not gonna be even one Drosophila person, that's a big deal, because you're right, like, yeah, it's a very different grant than if I'm writing to someone who knows about Balfour, subset, blah, blah, blah,
Yeah. And you have very little control over who the reviewers are. So you're trying to find that sweet spot. It's not like Jessica. Especially we did. So we had an A1. We had an R01. It was a MPI R01 that got well reviewed, some comments, put the A1 back in and it got hammered. And when I talked to the program officer and we looked at the panel, there wasn't a single, they were all animal modelers. And we were talking about studies in patient populations. So there was all kinds of reasons why they, like, I didn't know that's who I was talking to, right? I would have worded this completely differently if I'd known the audience. So it's tricky, but the person who's most likely to know is the specific NINDS program officer that's listed in the FOA.
Right.
Yeah.
Okay.
Yeah, so I'd recommend calling and talking to her.
Thank you.
And there are specific I don't like the word NOFO, but that's what they're supposed to be called now. The funding opportunity, there's specific call outs that the reviewers will see and their attention will be drawn to because of the peculiarity of the mechanism. So one of the first things the reviewer is going to do is look at this FOA and say, okay, well, I'm looking for this, this, and this. So doing things like calling out with specific language that you're addressing this part of what they're reviewing so they can find it easily is a really good idea, because I think my guess is that the DEIA part is going to be a big driver for the score. So, I mean, it just makes your life harder because everything else has to be perfect at the R01 level. Plus, you have to have an innovative DEIA plan or engagement plan. So I would talk to them as soon as you can get some time on their books.
Okay.
And the fact that there were representatives from NIH that came to Dell State I don't know if it's they would be very interested in talking to you also. Like you said, you are the person they're trying to write this thing for. So back and forth with them would be great.
Thank you. Thank you. Thank
Yeah.
you. Yeah. No, and I wrote that down because I agree. I mean, I've served on a study section before, and I mean, we're not supposed to talk about what happened in study sections, but
No,
I
can't tell
I
us anything
understand what you're
specific.
no, I said nothing. I said nothing.
Yeah,
Okay.
it's Wild West. You know, some of the established study sections, they develop a culture, and you know, you need to know people that have been on it. Like, but the people that can talk to you about it, because they're not part of the merit review or the program officers. They often listen in, they often sit in, even if they're not in the room, they're listening to the conversation. So they know what happens. And they're also the ones that inform how the funding opportunity is written and what they want to fund. And so they're a wealth of knowledge. But I would talk to them.
I'll email this week and try to get at least maybe even 15 minutes will do a lot just to get in that.
I bet they want to talk to you
on
for more than that. I bet they would want to, right? Because think about what the program officer does is they develop a portfolio of studies. They cannot control what CSR does or studies section or all the vagaries, but they have things that they want to fund because it's part of their portfolio back at the Institute and with council. So they have things that they want to support, whether or not you can get it. Because it's a separate arm through CSR, through the scientific review, is a different thing. But like you said, looking at the reward mechanism and kind of what you work on, what NINDS in particular is looking for, I think you kind of hit a wheelhouse for them. So you might find that they want to talk to you for more than 15 minutes. I wouldn't, it's part of their job is to get their portfolio stronger with these kinds of applications. So any other thoughts from the folks on the call that you?
One other thing that I'd written down that I don't think I said, I feel like you really do have some novel aspects to this proposal, but they don't come across. And at least in recent reviews that I've gotten, they're really looking for novel approaches and just I think it's there. I think it's just a way of writing about it to convince the reviewers. You know, for example, like the sleep, that just even looking at that is very different than what most people are doing, but it needs to come across that way.
Yeah, no, like I said, I'm very, very thankful. Yeah, I can't thank you enough for this, especially this format, because it's like there's what goes into the writing, right? But there's also, you know, the kind of input I would have to then put it in there. It's huge. Thank you. I really appreciate that. I have a couple of questions for you. One is what's not apparent in this aim space, because I didn't have space for it, is I have a couple of collaborations that are going to power this. Part of this is cynical, to be honest, but part of it is also I need it, right? So the sleep part, I have a collaboration with my colleague at UCLA. He has an entire room for sleep, right? No, sorry, I said that the wrong way. He doesn't sleep in the room. He does sleep studies. So that's already powered in too for me. And so my question is, to what extent should I highlight that as opposed to work that will be done at Dell State, right? So I want it to help the grant, not hurt it, I guess is my.
So I can
Do you have
comment on that.
preliminary sleep data? Oh, sorry.
Yes.
Well, go ahead.
You
Go ahead
do?
with yours.
Yes.
Yeah, so I think I was just going to ask if he had preliminary sleep data, and I think that also that often, like, the proof is in the pudding, right? Like the data kind of speak for themselves. So I think if you have that data in hand, it's fine. Like from my perspective, from my R01, I got a letter of like support from Josh Nunabel because I hadn't doing some ultrasonic vocalization stuff and I didn't have any experience with that. So I got a letter of support from him just saying, I'm here, you know, so you can get a formal letter of collaboration or you can get like a formal or like a less formal letter of support that sort of says that you have
Yeah.
somebody who's an expert in that thing if you feel like that's going to be that's going to be picked up by a reviewer as something that's potentially a weakness.
Right. And so I'm actually even want to go further than that. Like, should I grant them effort on the grant? Like, should they get, like, I don't know, 0.5 months, versus like you said, a letter of support? Because right now it's a pretty solid collaboration. Our folks have gone to their lab to run experiments there. That's how we got that data. But I'm also kind of mindful that, it's really a balancing act, right? Especially because of this particular mechanism, like Rob said. So I don't really know.
I think that, right, there's all of the different levels, right? So maybe, as a collaborator, like half a month effort or 1 month effort, would be fine. You're obviously the lead PI on this. Most of the work is being done in your lab, but that shows an additional level of commitment and collaboration. You still need the letter of support from him. So
Right.
even if he's receiving effort, you're still going to need the letter of support.
Right.
I also think, right, that would help your overall. I know all the scoring for the NIH grants is changing now. So, right, these don't get the necessary scores, but it would increase your score of whether this is feasible or not. Right, they want to make sure that you're communicating with other people in the field, that you're not an island. So I think that's fine. I also think that in the innovation section, that's a place where you could really, again, I think it was Amanda who said, like, mention things multiple places, but not just to mention in the research plan section, but also like the innovation section as well, that there's this collaboration to do these new experiments with sleep or something. We've already worked together. I don't know.
And you could lean into the PDP, like, so that there's a plan that you have to put in place about which, in that specific part of the funding opportunity, it asks about diverse institutions.
Yes.
So you can actually lean into the fact that you're an expert in inclusion of diverse, underrepresented students, and go the other way. Help.
Oh, wow.
Yeah.
Yeah.
I mean, I think that would be huge for this mechanism. So it's a true collaboration. He's helping you use that more established sleep. You have very specific outline things.
Yeah.
So I'm trying to read Diane's comments too. But lean in. I would lean into the DEI.
I hadn't thought about that.
I think,
Rob,
and I might be, once you talk to the program officer, they might say, no, don't do that. But from my understanding of the mechanism, that's a big part of how you're going to be reviewed. So if you've got all the technical parts, nailed, and in addition, in the letter back and forth, your colleague recognizes, okay, well, I could use some help with this. And in fact, we can have some joint programs, and you can help expand your experience with inclusion to UCLA, right? That's a good idea. Yeah.
And Hakeem, you said you'd sent students out there before. Have you sent students to UCLA to his lab?
Yeah, multiple times. Yeah.
Yeah. So that's I would agree with Amanda there in the chat that should be highlighted, because look, now your students are going to like a new environment. This is giving these students more opportunities to make more connections that could lead to them being a graduate student there or a postdoc there, right? Depending if you're sending undergrads or grad students, right?
Both. Yeah.
So.
Yeah, that's great. So we are at the end, at least for me. I have another meeting I have to jump to. I am happy. I think this went pretty well. It's recorded, so Julianne can get you the recording or a way to access it. And I'm happy to look at additional edits or talk more if you want to. Just let me know. Remind me, what's your submission target? What
May 5th. May 5th.
Okay, so you have a little window. That's good.
Yeah. So Okay. Awesome.
And reviewer, everybody, thank you so much for doing this. I think this is a really helpful, and from my perspective, both ways. I've chaired problematic study sections, special emphasis groups, and I really appreciate the collaborative effort here, because sometimes it's really not that collaborative when reviewers get to see things, as we've all experienced. So I appreciate it. And, you know, I do these also. I don't organize them through Julianne, but I recommend it. You know, it really is extremely helpful, especially if you're targeting a new mechanism, or you're going to a new institute, or there's something, it's not your study section that that you're familiar with, because getting eyes on, and especially, Diane mentioned this at the beginning, especially people who are knowledgeable but aren't really in the field. Because if you can get them and they understand it and they can follow everything, and then you get some reviewers who are very specific about exactly which model should you use and which molecules and pathways you should be looking at, that's really strong. So I hope you guys will participate more going forward. But good luck, Hakeem.
Thank you.
I'm going to have to
Thank
log
you.
off
Thank you.
now. And thank you
I'll
guys.
keep-
Thanks everyone very much.
Thank
It's
you.
very
I'll keep you
helpful.
updated. Thank you.
Thank you all
Yeah,
so
thanks.
much. If anyone has any written notes that they'd like to share with Hakeem, please send them to me and I'll be happy to compile them into one email for him. Thank you all
Hakeem,
so much.
I'm gonna summarize
guys.
everything and
Thank
send
you.
it.
Much appreciated, everyone. Take care.
Yeah. Bye-bye.
Bye.